pause for just a second and remind the folks who are joining us online that if you have questions, just type them in the chat. We are monitoring the chat, and we have someone here who is going to convey those questions to us live. So I just want to say that. And with that, Jim, why don't we start by you kind of sharing with the group here today exactly what MDI Bioscience is and what is the intent? What's the goal of this program, and how is it relevant to us?
Sure. So, when you think about bringing a new pharmaceutical to market, it's a remarkably complex process. And most people really think about the clinical side, where you're exposing humans to new drugs with the anticipated outcome being that there's a clinical benefit and the patient feels better, or it ameliorates a disease or whatever it might be. But where we're coming into play is actually before any clinical exposure happens. And the part of the drug discovery continuum that you don't really hear much about is how did it get to the clinic in the first place? And the fact that 1 in 5,000 drugs actually makes it to market is pretty astounding. I mean, I don't really know any other businesses where 1 in 5,000 products can actually succeed and you can still have a business at the end of the day. So a lot of what you're trying to do in pharmaceutical development is actually de-risk the compounds that you're trying to develop. And what I mean by de-risk is you're trying to figure out whether they're toxic or whether they have the intended benefit. And if you can do that earlier in the process, if you can really figure out what you have right out of the gate, it sets a positive trajectory for the rest of the program. And that's something that I don't think we do particularly well right now. And that's because we're oftentimes evaluating new compounds in single cells. And while single cells have had an incredible benefit to advancing drug discovery, it has also omitted what I think is incredibly important in biology, which is systems biology, which is the interconnectedness between all the organ systems. And so what we're trying to do is we're trying to bring MDIBL's expertise in non-mammalian models such as zebrafish and C. elegans, and we're trying to bring that whole systems biology way, way, way earlier in the process so that you can actually combine cell biology and systems biology earlier in the process using what we call high throughput screening, which means taking many compounds and being able to screen them against a particular type of genetic model that may represent a disease or a genetic mutation. So that will really, I think, bring a lot of robust characterization to new compounds going forward, such that we will understand better which compounds are toxic to non-target organ systems. Everybody focuses on the organ that you're trying to affect, but oftentimes what happens is you have an undesirable effect on an organ system that you didn't want to impact. And so the sooner we can get to know, the sooner we can fail, which is where the origin of this talk came from, the title, the better off we all are, because it allows us to focus on the drugs with the greatest potential.
That's great. So, speaking of kind of failing or being more efficient with how we do this work, I know that certainly has been kind of the mantra here at MDIBL for 100 years, because quite honestly, out of necessity, right? We're a relatively small institution, kind of scrappy, as we used to say, sort of holding everything together with a little bit of duct tape and baling twine. But we're now progressing, I think, to a point where we have potentially an opportunity to explore some of the ways that this sort of approach to science, if you will, can really be applied. And Dr. Howe, that's what I was curious, your take on sort of where does this fit in MDI?
But Terry, first of all, I mean, you have to change these words. I mean, if Tim wants to be successful, duct tape doesn't work in the pharmaceutical industry. But I think there are two messages which are very important. First of all, and Tim already mentioned this, we are bringing our expertise to drug development. So the question is, does this change MDI? Yes and no. No, because this is only possible on the basis of our basic research. To be honest, we have grown a lot. We are much better than we were a couple of years ago. We are now an institution with a lot of very original and very strong basic research. And this is the foundation. We can then have something like MDI Biosciences on top of it. So yes, it will reflect back on what we're doing in research because it's translational. It opens our research. We did this before, but now in a very different way, we open our research to this translational aspect, to drug development. But the strength of MDIBL is the basic research. The strength of MDIBL is the communication, is the student, is the researchers, is the visiting researchers. And this is what we have here on campus. And then we add to that quite an important addition. And we're very lucky to have Jim Strickland with us. Krista was asking, are others doing the same? Yes, but without Jim Strickland. So, and without our strength. So what Jim has just outlined, that we now want specifically to add non-mammalian animal models, one of the words Jerry likes the best. Non-mammalian animal models is between, and I think Jim will go into this in a moment, between this cellular approach or molecular approach where you identify, oh yes, perhaps this compound is working. And then you go all the way to the next animal model, which is mammalian, which is the mouse. And in between, there's nothing. A lot of time, a lot of dollars, a lot of effort are really spent in between. And here it is where we come with our expertise. Now, like Dustin, who understands C. elegans, we couldn't do this with our research on C. elegans. Ian, with his expertise in zebrafish, yes, we can claim we're one of the most important basic research labs, at least this side of the Missouri. And we can, well, I chose this by accident. Sorry about that. So it will be fascinating to see now how this works. And Jim and I, and Ian as well, we were in Boston. This was only 6 months ago, less than 6 months ago. This was end of February, where we presented this for the first time. And I can tell you, we're spending now a lot of time talking to biotech, talking to pharmaceutical companies, and we learned from them as well, because all of a sudden there's more of a discussion going on how to use us in biomedical research, and we learn from them as well. We start with some of these companies and we discuss very fascinating approaches. Last week, we talked about killifish, our aging model, and the nervous system, and we end up with zebrafish and aging, and this is because of their input into our research. So I'll stop. Sorry.
Well, so the question that comes to mind for me is, you have an industry that is kind of ripe for improvement, shall we say, or in greater efficiency. Why are they not doing this anyway? Why is this not the approach that they're taking? Why are they not looking for a more efficient model to kind of speed things up? I'm just.
Excuse me, repeat that. The last part of the question. Why?
Why is the pharmaceutical industry not doing this on their own? I mean, why are they not doing this, or have they not found another partner to do this? Why is there a unique opportunity for MDIBL in this case?
Excellent question. And I think part of it just comes down to, we all get up in the morning and we do kind of what we did yesterday. It's the tools they know how to use. It's the machines they have. It's they're looking for ways to do better, but oftentimes they want somebody to show them how it's done better. And I think that's where we come in is, we've got all the expertise on the animal models here, and that's not widely available in industry. So, what we're doing in a lot of these meetings is we're teaching on how the animals actually, are representative of whatever molecular target or disease that they may want to address. And I think that's really where we're adding a lot of value. And I think it's where our pharmaceutical partners are starting to say, there's something real here. Show us, show us more. So.
Question?
And just one brief
On the Zoom, who may not have heard.
So I think the gist of the question is, what are the advantages to doing the work in zebrafish as opposed to other models, including rodents and other models like three-dimensional organ systems like organoids. So I'll give you my answer and then I'll let Professor Haller provide his, but the ability to do high throughput screening is a huge potential benefit. And I think organoids and zebrafish and C. elegans offer you the ability to look at huge compound libraries quickly. And so that's certainly an advantage over rodents where you really can't do high throughput screening. And then the genetic homology of the zebrafish to human disease causing genes is something like 85%. So it's remarkably similar to humans and its genetic makeup.
So I just want to go back for the non-scientists in the audience. When we talk about these high throughput screens, can you just explain what that is for us? And
Sure. So lots of times when you're trying to find a drug that is active against a disease, then lots of times you start out with basically structures or different types of molecules that you think might have the potential of acting on a protein or a specific target within the body. And there are compound libraries. Lots of times they're naturally derived compounds, just huge numbers of compounds and derivatives that you can actually, either they're proprietary to a company or they have the ability to buy them from a third party. And so what you're doing is you're trying to test as many different compounds as possible against your disease model in order to see which of those compounds has an interesting effect.
And we're talking about thousands of molecules you make tests. But the question also addresses much more basic questions of drug development. Because as Jim pointed out, most of us do tomorrow what we have been doing yesterday. And in drug development, it's the same thing. 50 years ago, mice didn't exist for drug screening. It was rats and dogs. Imagine dogs today, that's out of the question. So the question is, what is a model? And this is what we are discussing with the pharmaceutical industry, because to be honest, a mouse is not a good model, disease model for humans. Most kidney diseases we have in the mouse do not reflect what's going on in humans. We still have to use them. We have nothing better. But the question is, again, what is a model? So we are using our model to genetically tailor diseases. And at the moment, this is all the rage because we now recognize that it's not only histology. When we look at the tissue, it's more about genetic mechanisms of disease. And we have with our models, we are able to make genetic models for the disease. But this is educational. It's not that it comes easy to a lot of these pharmaceutical companies when we talk to them, but then we start very interesting discussions how they could change their models. And again, here comes our expertise. Because then all of a sudden we are serious partners to use these novel models because yes, they heard about superfish 10 years ago. I mean, it's not our own invention, but we are serious and we are discussing with them, and this is a very important area. And she was very experienced with that, to take them seriously with their questions and then translate this into new models.
Yeah, I think,
I think we should repeat the question.
Yeah,
again.
so the question was, does that MDIVL have expertise in high throughput screening? Is that essentially, I mean, where?
I think the short answer is yes. And the microscopes that we already have are plenty capable of doing high throughput screening. And I think there have been research groups here at MDIVL who have done high throughput screening. And so it's not something they do every day, but it's something they know how to do. And I think it's a skill set that we're going to be growing over time as well. We're right now demoing a high throughput microscope from Europe that is really phenomenal and uses artificial intelligence to actually look at the different pieces, organs of a zebrafish, pieces, and actually identifies the different organs and looks at the fluorescence of those organs and model systems. So it's a really important part of what we're doing.
And this, your question actually goes beyond that, because it asks the question, are you up to it not only because of screening, but data safety? Reproducibility. I mean, the pharmaceutical industry is not giving us contracts if they are not 150% sure that the data are safe, that the data are reproducible, that our data management is on the highest standard. And for some of you, when you come on campus, you may ask, where are they actually doing this? And this is a wonderful example of old and new. We are restructuring at the moment the Halsey lab, so one of the lower campus lab, and mid of August. We originally thought 1st of August, but mid of August, we'll have their first-class facility where when we invite international companies here, we can show them very clearly that MDIVL is not only of high throughput screening and our expertise, but also in term on the same level as the pharmaceutical industry to do this one.
Dr. Hall, I'm going to have you move your microphone down. It's putting on your collar, apparently causing some feedback. Sorry. Are there other questions from the audience? Oh, we have one from someone online. Jim?
It's a hard question. You know, it's a very good question. The question is, how much validation have you already done? Right? The question is, how many models do you have? How many models you have generated and you can use? And we're working at the moment. We have models, but to validate the models, to make sure that they have tested against other already existing drugs. That's what we're doing at the moment. So it's a very good question, and it's a lot of work, but we are validating, especially our new models, and then we can go on the market with them. Well, with all drugs, it's not only about gene therapy. In fact, it's not that much about gene therapy in the first place. We are looking into, so to speak, normal drugs, normal compounds, but the models we generate are more genetically defined disease models. You may ask yourself, what do you mean by that? We are in an era of rare diseases, and these rare diseases are genetic diseases. We have a lot of kidney diseases where we have realized for some of the diseases, when I was a young doctor, they were called focus sclerosis. And now I know that there are 153 different genetic alterations with focus sclerosis. So when you now look for drugs, you better not look under the old classification, but you understand what's going on. And this is why we all of a sudden can then generate a whole bouquet of genetically altered animals so that pharmaceutical companies can test it.
Are there any diseases or types of disease where models that we would generate would not be applicable? I mean, I think the answer to that is we've already said no to a number of projects where we just didn't feel like it was the right fit for the type of therapeutic that was being developed. But I think that there are many more avenues where it is the right model, and there isn't a species out there you could say that about. So.
We have one more from online.
So the question is this more of where we're looking to develop our own drugs on a sort of de novo basis, or are we looking at working with industry to essentially help them with their development programs? And the response is that it's predominantly the latter model, where we're plugging in our expertise into pharmaceutical companies' development pipeline and adding sort of a turbo boost to their discovery program so that they can get the right drugs prioritized in the pipeline.
And we know that this will work. I've started a company doing this exactly in mind 20 years ago. And when you have the technical tools, when you can transplant organs, when you can operate, when you can use your animal models, then this is important for small biotech companies because they don't have the means really to build their own model. And the large companies, when they have programs and they want to have things done very fast. So that's the start. However, having said this, some of our scientists come up with a brilliant idea, and we find compounds and we patent them. Then this initiative is here to develop this further.
Great question. So the question is, has the development of COVID vaccines led to sort of a rethinking of how pharmaceuticals are developed and brought to market? I think unequivocally the answer is yes. Everybody's thinking about it. How many people are actually doing it is the next question. And I think as we look back on what's happened with the development of the COVID vaccines, a lot of it was due to an unprecedented funneling of resources in one specific direction. And that can't be replicated. We just can't do that for every single disease. However, there were lots of new approaches and new technologies that were used throughout that process. And I think it got the attention of the regulatory authorities and thinking about where are the processes and what laborious things need to be. The FDA standard is that the process of getting a drug to market should be no more burdensome than science requires. And so their task actually was trying to make their processes more efficient. And I think that's where we're going to see the greatest benefit is the sort of learning from the regulatory perspective on how efficiencies can be made.
And the FDA is also part of this. We have not only talked to biotech and pharmaceutical companies, but we have to talk to the FDA. Which models do you accept? And are you willing, and we are much faster, that the preclinical process before you go into humans can be sped up by our work considerably and only 50% of the lifetime. So they have to be protocol and they are protocol that we anticipate legally with the FDA at MDI discusses the implication of using different models.
Most people don't realize it takes 15 years to get a drug, the lab on the market, and it takes last estimate is over $2 billion. So it's an incredibly laborious process, and you can imagine that if you get into year 7 or 8 of your development pathway and find that there's a toxicity or it doesn't have an intended effect. That's a tremendous amount of time and resources that have been devoted to a path that has no future. So that's what we're trying to do is to help people recognize and characterize their work lately to avoid the number of drugs that get that far in development and fail. So the great thing about zebrafish is they're prolific, and so generating enough zebrafish. We always worry about having specimens, but at the same time, zebrafish are pretty prolific in their ability to generate numbers for studies. And the second part of that question was. Yes. Okay. Thank you. So, C. elegance are so used in drug screening, and so there's tremendous potential for the use of C. elegans in drug screening. I think the axolotl we have it's very fish, but also have really unique properties that make them potential models for drug development, but they're not as far along as zebrafish and C. elegans in terms of industry recognition of the viability of the models.
But even at these early stages, they resemble humans to a large extent. We have these tiny zebrafish embryos. They have one glomerulus, which is the essential part of the kidney. And when you look at this under the microscope, it looks like a human glomerulus. So they have a high homology, so they don't look human, but the structural and the functional properties of these animals are very useful in order to look, as you mentioned in the beginning, are substances toxic, or do they actually have a beneficial effect when you have already altered the fish? So we can make these fish, for instance, diabetic. You were asking the question, which diseases? So yes, we are concentrating at the moment on kidney disease and diabetes. Diabetic nephropathy is the leading cause of kidney disease, leading cause of dialysis and transplantation in the US and worldwide. And we can make these tiny zebrafish diabetic, and then we can look what damage the diabetes does, and then we can screen for drugs. That's what we are trying to do, and this we can do in large numbers. Tim said they are very fertile. Yes, they are, but we never have enough zebrafish.
And so in terms of, number one, we've talked about, we have a transparent model so that we can actually watch development happening in real time. What does that timeline look like? How long from kind of fertilization until it is a fully formed?
Ian, how long does it take before
We can see a kidney.
2.5 days, so they have all functioning organs at 2 days after being fertilized.
So that gives people a sense of how quickly you can watch, this process happening. Okay?
What a gigantic thing for students, I mean, when you work with mice, it takes forever till you have a grown-up mice. I mean, here you can actually do the experiment within 1 week.
Definitely speeds things along. Any other questions from the audience or from folks online? Go ahead.
A very basic question. You use the word model. We understand that with the mRNA development so fast in science, it was done with the use of technology. Can you give an example of a model, a very specific model, bringing it down for non-scientists?
I try. I was already warned that we'll go down to Socrates and really philosophical question. So the question about a model, and I give you one example. And I mentioned this before. When we classify diseases, and I'm a medical doctor, and I'm a kidney doctor, and I have words for diseases, and most of these classifications come from the microscope. So we have started to biopsy kidneys about 60 years ago, and now we have a full classification from the microscope. And now it turns out that this classification is perhaps not incorrect, but you need other models. We find that we have genetic alterations and they resemble each other, but very different molecules are affected. So all of a sudden, the model is not a model of what you see under the microscope, but the model is a genetic model where you change one gene and then you get what we call a phenotype that you can see this disease. This changes almost everything because then it becomes very personalized and we care about that. Personalized medicine means that we have to understand the genetic makeup of individual patients and to have models for that, which we also need because we can't treat patients all the time and just by experience, and we have to understand what's going on. This is what we would call genetic models of disease, and here the zebrafish is an ideal animal model.
Great. Wonderful. So Jim, you told us that, you have this interesting idea. We feel as though the time is right for a number of reasons, right? We have an industry that maybe could use a little help in improving its efficiencies. We've just come through a pandemic where we've seen kind of this real life in real time example of how speeding along these sorts of treatments or preventative measures are so important. Tell us what the next phase looks like for you. So where do we go from here? You've had some interesting conversations with members of industry. What's next for you in this project?
So I think, it's been an interesting evolution in that, as Dr. Holler noted, we've been learning as we've been going along. As we have these conversations, we're really taking notes as we get feedback from our pharmaceutical industry partners. And we're refining what we're doing all the time. And so I think as we look to the future, what we'll do is we'll be aggregating more technologies that fill specific roles in a particular development program where they may fit for program A, they may not fit for program B, and vice versa. But there's tremendous opportunity for the predictivity of these models and the ability to generate efficiency using these models to become even greater with the use of artificial intelligence and data science. And so we're looking at ways that we can create some of these combinatorial benefits of taking different technologies, marrying them together, and bringing them to market in a unique and novel way that may actually improve the ability of the models that we've been talking about to determine whether a drug has efficacy against a disease or may have a toxic effect.
Mm-hmm.
And from what you've just heard, you can see how many avenues this opens for MDI. So you were talking about bioinformatics, you were talking about modeling. We are very intensively talking to the Ru Institute because with the new Ru Institute, there's 300 million in program, but they have tools. And we have the problems and we have the model. So we collaborate with our approach. You can imagine that we are interested in kidney disease and other diseases, but the cardiologists in Portland, they are talking to us. And the Institute of Medicine in Oregon, they are interested in, oh, what's MDI they are doing there? They have new models and we could collaborate. So all of a sudden, MDI who is already associated and has lots of collaborations within Maine on another level makes a big difference.
That's great. So you'd hope to have a renovated facility in a couple of months or a month and a half, but about right.
Mhm.
And so then is it really an opportunity to just sort of have proof of concept? You know, we talked about validation, like how does that move forward in the next
Yeah.
phase?
So I think, you know, as with a lot of projects, you kind of want to make sure that you get it right first.
Yeah.
And so we're really focused on getting one or two initial projects through the door where we can closely work through our standard operating procedures, our security, you know, all the different elements that have to kind of come into you know, come together for a project like this to work and for pharma to really adopt it. And so I think that's really sort of the next 6 to 12 months. That's really where we're going to be focusing on. And these are long projects. They're long to develop and they're long to execute. So, you know, we may be working with a pharmaceutical company for a year or two to get through a specific project. And so I think we're still in the learning phase and we're still sort of, you know, really getting our sea legs under us, but it's an exciting period. It really is to be able to see the industry that sort of, and their response to what we're bringing, I think is heartening and sort of confirms that we're on the right track.
That's really cool.
And you never know where this journey will lead us. Now, you look at our neighbors here on the island. I mean, nobody would have thought 30 years ago that the mouths of all animals would be the experimental model for the pharmaceutical industry. And some of you may remember the tax was not always as big as it was. So it may eventually be, although we are small and beautiful, so we don't want to become like the tax, but nonetheless, we don't know. We don't know how this will develop. However, we think it's now the age of non-mammalian animal models.
The tax is beautiful too. We love them. Don't be worried. Mhm.
But there are several answers to that. First of all, yes, I think openness is wonderful. And yes, we'll stay like this. But you may have realized MDI has changed over the last 20 years. When I first came here, it was much more relaxed and not everything was good. But we are a little bit more secure. Our animal facility is more secure. So this is development when you're doing serious research. The next level, however, and we have discussed this very early on, we need this level of security, but we don't need it everywhere on campus. So this will be confined. Oh, don't worry, we won't block you away. But this will be confined to special areas on campus and not change MDI completely.
And I'd like to add to that too, that we're offering an innovation course this summer where we have the opportunity to work with the students on campus where they get maybe a real world perspective on what does it take to get from bench to bedside with a new pharmaceutical product. And I have had the time of my life teaching these kids because it's been the feedback and the questions that I've gotten are so incredible and show a level of engagement that is beyond what I hoped, quite honestly. So.
And this is also, I forgot about that. You know, I was teaching at Kobe about biotech, but what Jim is now doing with this new course, first of all, we have lectures from outside. Your friends from pharmaceutical industry and biotech, they come and lecture there on Zoom. And this course is now of a very high quality for Colby students as part of their biotech program and also for other summer students. So this is something very novel. Jim is very engaged and lots of critical discussions, lots of very open discussions. Is wonderful.
But to your point, that is something that we think a lot about, right? So MDIBL, as Dr. Haller said, over the last 25 years, I mean, I was the 9th full-time employee that came to work here. We now have 85 employees. So, I mean, there has been a tremendous amount of change. But we have to be intentional about holding on to those things that we know set us apart, right? And that we know foster that sort of creativity, that exploration, that spirit of collaboration. And so that's going to be the challenge for us. And I think that's where we can come together as a community and speak those things and continue to kind of live that culture and those core values. I think it's going to be incredibly important. And I think we can create a space for both of those things. And as long as we name them and we actively work to protect them. And I think that's one of the things that really would be holding up to us to make sure that we do that.
And the guarantee are the people working on. It's not only Terry and all the others here, but for instance, the HR. Look, we do it by the book. But nonetheless, we are still a place where you can openly discuss problems, and we're still in the idea. Book is responsible for.
Send inquiries over there. Well, listen, this has been a really fascinating discussion, but I want to close with this comment, and you know that I'm the one who's responsible for fundraising, so I can't let this opportunity pass us by. But to really highlight, the role of philanthropy in bringing ideas like this to fruition. So without the support of donors, like many of you who are here with us this afternoon, we wouldn't be able to explore these ideas fully. It allowed us to bring Jim on board. It's allowed us to make these renovations to the laboratory facilities. And so it's a really clear example of how those kinds of funds really allow us to pursue something and then potentially develop an entirely new revenue stream for the institution going forward. So I just want to say thank you to those of you who see that opportunity and you're willing to invest, because that's really what is so critical in moving the institution forward. And it plays a really, really big role. So hats off to all of you.
Terry, may I add something?
Sure.
It's great that Jim Strickland has joined us. Terry was just talking about MDI values and all that. And Jim, thank you very much. Wonderful member of MDI, and it seems like he has been here for the last 15 years.
I want to just remind you of a couple of in-person events that we have coming up. So next week, we will see the return of Family Science Night. So it's been a 2-year hiatus for that much beloved program. So we invite all of you to come back to campus. You don't have to have a little one. Everyone's invited. It's a great opportunity, not only to see some hands-on examples of the research that's happening here, but to meet our students. So the way this event works is we task each of the students that's here on fellowship to kind of create an activity, sort of ages, say 5 to 12, but we let adults come and we don't tell anybody, it's fine. And we ask them to sort of convey 3 main points about the work that they're doing this summer. And I have to tell you, this is my favorite event all year. It's so much fun. And we turn the whole lower campus into essentially an outside science fair. And there's everything from a beanbag kidney toss to finding your way through a kidney maze or learning about environmental programs. This year, we actually opened our campus to some partners. So we're going to have folks from the Landscape of Change project, which is a collaboration between 4 or 5 entities on MDI, including the MDI Biological Lab, more of a hands-on citizen science program. We want to make sure folks are aware of that, as well as our friends from the Maine Discovery Museum will be here with some activities. And we have, for the first time, a returning MDIBL alum who's coming back to have, she's developed a little project to help support folks in more rural areas of the state to be able to come and participate in science. So she's really interested in STEM education and wanting to reach out to the community with that. So I encourage you to come back July 6th, 4 to 6 p.m. And then we will have our regular host of Science Cafe events every other Monday. So we'll be back here. And all of that information is online on our website. And I'm sure those of you who are on our mailing list will be getting more details of when and where all those activities will be happening. So